Okay, that's small molecule drug design and a backdrop into what leads us into DNA-based drugs. And when I say DNA-based drugs, I'm referring specifically to those compounds that are derived from recombinant DNA. Remember in previous lectures I have described how we can now clone any piece of DNA. And we can clone the pieces of DNA which regulate whether that gene is expressed or not. And we can fuse those elements together. And we can put a termination signal on there and introduce that into other organisms that will still synthesize that same protein off of that foreign piece of DNA. So, for example, I brought up the case where we could clone the gene for your hemoglobin, a blood protein. And I could put it behind a promoter or regulatory sequence that would be recognized by a corn plant. Similarly, a compatible termination signal, I can introduce that into a corn plant, and the corn plant will, lo and behold, make hemoglobin that would be identical to yours. I don't have any good reason to do that, but that has been done, actually. Human hemoglobin has been produced in plants, along with lots of other compounds. We can take the same technology to make insulin. Insulin was, in fact, the first cloned drug, cloned gene drug that was produced using that technology and widely applicable. So what are the implications of gene cloning now? Well, like my example with insulin, you can take your favorite gene that is protein-based, drug approach, and put it into a cassette that would look something like this, introduce that element into the appropriate cell source to produce that protein. The cell source in this case could in fact be plants. We can make pharmaceuticals in plants. We can make antibodies and vaccines in plants. But more than likely it'll be a mammalian cell system. It could be a yeast cell system. It could be in fact a bacterial cell system. DNA is DNA. So drugs like Enbrel, for example, produced by Amgen to treat rheumatoid arthritis, and I'll talk about that in a little while, or the drug Herceptin that's used as an antibody treatment against certain types of cancer, and some of the other monoclonal antibody drugs that are on this list. And that's a short list. But these DNA technologies as pharmaceuticals will now allow precise treatment of diseases because antibodies and these drugs will target specifically, chemically, their target without side effects. Rather than using a drug and a chemotherapy which will blanketly immunosuppress a system with huge side effects, we can now target specifically receptors and proteins on the outsides of certain cells like cancer, or in fact certain microbes or invading viruses and so on. This is a much more intelligent and articulate approach to drug design than the spray and pray approach that we came from in the past. So this kind of an approach leads into biopharmaceuticals, and largely big pharma, no matter what the company is, has taken hold into these protein-based therapeutics because that's the future. If you're not on that bus, the bus is left, and you're no longer competing with the big dogs. This all owes to the fact, like I said, that DNA is DNA, and the genetic code is the same virtually in all organisms. So the genetic code, as shown here on this slide, indicates that we have 20 amino acids, and the genetic code is the same for those amino acids no matter what the organism is. So if you look at this code here, which was originally devised by Nuremberg and Matthiai in 1962, doing simple experiments on phenylalanine, you can see you want to make two observations. The code is redundant, and it has start and stop signals for all amino acids, all 20. And you might ask the question, why are there 20? There are more than 20 amino acids in nature. Why do all biological organisms use 20 amino acids, and they use the same 20. There's some that are more abundant than others, but it's generally not 21, or there are some that do use 21 or 22, but there are some weird amino acids that some organisms use, but generally speaking, 20 is the number. And if you look at these, the way you read this chart is on the left, you look at the first position, say we'll call that A, and at the top you read the second position, let's call that one U, and on the far right you look at the third position, let's go down and make that G, then you can use your finger and go over and you see that brown box in the first column there is methionine. So AUG is methionine. The reason I bring that one up is, one, that's the start signal for most genes. Most genes in nature start with AUG. That's pretty cool. So you have a start signal. Then if you look at this, let's take UAG, you have stop. So you have a start and a stop signal. And you can see that other ones like CUG gives you leucine, but so will CUC give you leucine. So the code is redundant for some, and it has starts and stops, but information is the same in all organisms. So if I take his hemoglobin gene and I put it into a corn plant, the corn plant will make hemoglobin. If I take his hemoglobin gene and put it into a Chinese hamster ovary cell, that Chinese hamster ovary cell will make hemoglobin, or a yeast cell, because the genetic code for that hemoglobin is the same for those amino acids that's spelled for that protein. No sweat. There are codon preferences that are tweaked, but basically that's the way it goes. So we can take these genetically engineered cells, say the code for the antibody that's used in Enbrel, and we can put that into a Chinese hamster cell as our cell of choice, which will fold that protein accordingly because it's a mammalian cell source. And we can make a master cell bank that we can use to inoculate a culture, grow it in production in a large fermentator, harvest those cells after they've reached exponential phase, and we harvest and concentrate the cells, purify out the protein, and then freeze dry it, that is, lyophilize it, and sell it in a vial as an injectable drug like Enbrel. And the reason it's injectable is this is a large molecule. And if you were to eat the protein, you're just going to digest it like any other protein and it won't have any effect, so it's an injectable drug. This brings up the whole aspect of biomanufacturing. You go down the road from here and you look at the plant that Enbrel is made in by Amgen, and that's the drug that they make there. That is pretty much what they do at that facility. And the science and the engineering that goes into biomanufacturing is a whole career opportunity unto itself, and a whole situation that will be ongoing as this science continues to mature. And like many of these things, is outsourced to other countries that can do this more cheaply. China and India are heavily involved in manufacturing of pharmaceuticals. This brings me now to vaccines. Vaccines and our immune system. Our immune system, you know, makes antibodies. And vaccine development is intimately tied to antibody production. So I want to put those together right here, because DNA-based technologies are vastly affecting the way we make vaccines. And antibody production that's affected by vaccines is important to understand. How does a vaccine work? But also it's important to understand that we can now make antibodies or synthetic antibody fragments that they themselves can be used as drugs, like Enbrel, like Herceptin, like other monoclonal antibody cells. But this brings up all kinds of weird controversies. Controversies about vaccines and human health that, quite frankly, I'm getting a little tired of hearing, so I think I will respond to that now. I think what we need is myth understood. That vaccines do not cause autism. Vaccines will not compromise your immune system. Why should all children be vaccinated? I want to get to that after we go through vaccines. That it's not okay to not have your kid vaccinated. Can a vaccine compromise your immune system? If that's true, you better not go outside, because you are confronted with pathogens and foreign compounds all the time, every day. If you think that one more compound is going to compromise your immune system, then you just simply don't understand how your immune system functions. This is just the tip of the iceberg. But how does genomics influence vaccine development into the future? This will become very important. When we encounter our next pandemic flu, we hope that we're onto this fast. And we better have a really good way of dealing with it. And I say when. It's not a matter of if. But why are there so many broad public misgivings now about vaccination? Why do people think this causes autism? Why is that propagated through the media? Before we get into the broad public misgivings about vaccines and vaccination, I think first we have to understand something about the immune system. Immunology is certainly a large and expanding field that's beyond the scope of this course, but let's try to understand some basics first. What happens when humans are invaded by a foreign organism? One thing I can tell you, that for a human to catch a cold is definitely a bummer. There's no doubt about that. But for a cold to catch a human, it's by far worse, because pretty much most of the time, the human wins that struggle. So if you look at this diagram off to the left here is of the human foot, where it's been wounded. That's the nail sticking into the side there that has injected some sort of bacteria that is first recognized as a foreign invader by the phagocytes. Now, it's pretty much a tenet in biology that recognition of self from non-self is one of the first origins of life on this planet, and is consistent with most life on this planet. These recognition systems of self being okay, non-self being not okay, are actually very ancient. So the development of the immune system actually goes back molecularly to origins of cells and life on this planet. And it's very highly articulated in some organisms, such as the immune system in animals. And the defense system in plants is also quite exquisite. Pretty much all organisms have recognition and defense systems. I mentioned previously that the human is 24,000 or 25,000 genes or so, whereas a rice plant is 48,000 genes. Most of those genes are involved in defense. You can run away from some of your predators. A plant has to sit there and fight it out. So defense mechanisms are all over the place. In this case, the first recognition signal is picked up by a phagocyte, and then this is recruited by helper T cells, and eventually antibodies are generated, which are proteins which are capable of recognizing either foreign proteins or any other foreign substance that we would call an antigen. So antibodies then are proteins that are encoded for by genes, and immunotherapy uses antibodies as drugs. Typically these antibodies, these proteins, fold into a characteristic Y-like shape shown in this molecular structure to the right, but they are also broken into fragments or defined by different fragments called variable heavy chains, variable light chains, constant heavy region chains, and constant light chains. And together, these segments come together in a unique way that will recognize the antigen that's been presented, whether that's a bacteria or some other protein source. So you're making a unique antibody against any introduced antigen. And this antibody will remain present in B lymphocytes so that if you ever encounter that foreign protein again, your immune system is already established to recognize that. What I just said is the basis of vaccines. So you can make antibodies as drugs like Enbrel by cloning fragments or an entire antibody that will recognize a specific antigen. So these can be made in systems that are called monoclonal antibodies, and we can make these monoclonal antibodies as therapeutics. So let me talk a little bit about what these MABs actually are, monoclonal antibodies, because they're widely used in the manufacturing of drugs, and we can use these to target certain proteins that are involved in diseases like rheumatoid arthritis and how Enbrel works. So antibodies then are these versatile proteins that are capable of recognizing a wide range of foreign substances and foreign proteins or antigens. So these antigens could be responsible for pathogen specificity. Imagine that you have a bacteria which is presenting proteins on its outside surface that is recognized, and an antibody is generated against that, and it causes those bacteria to coagulate together and then is recognized by phagocytes and consumed and eliminated. So that is one part of the immune system, but the part I really want to focus on is that these variable chains are constituting the antigen binding site. So they are what is going to recognize any foreign antigen, and if we want to do that for biotechnology purposes, it's that region of the antibody that we're really mostly interested in. The effector functions that cause neutralization or agglutination that result from the constant chain may also be functionally important. And these are presented in a lasting memory in your immune system on the B cell receptors. So what is a monoclonal antibody? How does this work? This is a way of generating a cell line which will continuously and reproducibly generate the antibody, whether that's going to be a drug or useful for some other application, like a home pregnancy test. Works also on monoclonal antibodies, and I'll give you an example of how that happens. But first, look at this example here, where if we were to take human insulin and inject it into a mouse, that mouse will generate antibodies in its B cells, specific to human insulin. So now you have a mouse antibody to human insulin. So the mouse recognizes human insulin as other, no good, and mounts an immune response, and the spleen can then be extracted, surgically removed from that injected mouse, and suspension of the B lymphocytes can be generated out of that extracted spleen. Meanwhile, you have a culture of melanoma cells that continuously reproduce in culture. You can keep in culture for a very long time. These could be human or some other source, and you make those into a suspension and fuse them together with the B lymphocytes. Some of those B lymphocytes are generating antibodies, and you can grow these in a specific hybridoma medium that will cause the hybrid cells to continuously divide. And then the hybridomas, these cells, these hybrid cells between the melanoma cell and the B lymphocyte that contains the antibody for the insulin, can be selected for by those that are producing antibodies specifically against insulin. And then you subculture those Now you have a culture that can grow continuously, making mouse anti-human insulin. That antibody can be used for all kinds of things. So that's just an example of how we would make a monoclonal antibody using a mouse system, but you can make even secondary antibodies. So now let's say I took that mouse antibody and I wanted to make a secondary antibody. I could use that mouse anti-human insulin, inject that into a rabbit. Now I've got rabbit anti-mouse human insulin. And I'll show you these combinations become, I know it looks confusing, but these combinations increase the resolution of detection. And I'll show you in a minute why that's important. But in a previous lecture, I have described how we can use these nitrocellulose filters to blot DNA and RNA and recognize specific sequences. And that's called a southern blot for DNA and a northern blot for RNA. The southern blot for DNA was named after the man that discovered it, J.D. Southern, in 1975. And then when they used it to detect RNA, they rather non-creatively called it a northern blot. And then when you use antibodies to detect proteins, this is called a western blot. So we can run a whole protein smear out onto a gel in gel electrophoresis, blot those proteins to a filter of nitrocellulose, which will bind all of the proteins, use a specific, and let's say your insulin is in that smear. We can find just that insulin with the mouse anti-insulin antibody. If I fuse that antibody to a colorimetric dye, you get the resultant detection of that protein in that gel. And I can use that same system to isolate it. So if I chemically tag the antibody with a fluorochrome or radioactivity or something, I can use the antibody to specifically detect its target. Very useful tool. Why is that useful? It's been used all over the place. We can use proteins then to detect, other proteins. That is, we can use the antibodies to specifically detect and find their target and even precipitate them out. One of its applications that's broadly used is in terms of detection in these applications of these monoclonal antibodies is called an ELISA technique. And we can use ELISA, which stands for enzyme-linked immunoabsorption assay. So we've got an enzyme that's linked to the monoclonal antibody and is used to generate a colorimetric product. In this case, let's take a monoclonal antibody that is against human chorionic gonadotropin, or HCG. HCG is produced a short time after human fertilization, and this protein then circulates in blood and urine and can be detected exquisitely. And the reason why this works is antibodies are exquisite at detection, first of all, and it can find a very, very low concentration of its target. So the way that this works is you make the monoclonal antibody against HCG and fuse it, say, to a plastic substrate, a plastic well in a petri dish, or a plastic dipstick will work. And when it is exposed to its specific antigen, that specific antigen can be detected by a second monoclonal antibody, increasing its resolution, as I just described. And that secondary monoclonal antibody is fused to a colorimetric product. This happens very quickly, and you can detect HCG at parts per million, maybe even below that, to evaluate pregnancy. And this is what has been commercialized in home pregnancy tests, and we feel quite okay about doing that in a home pregnancy test, owing to the fact that it is extremely efficacious and reproducible, even in the hands of a home practitioner. Okay, that's a brief primer on the immune system, but we can generate antibodies against things, all right? And animals do that. And these antibodies are specific against their antigen targets. And this is really what brings up the basis of vaccines and vaccinations. And I want to, first of all, describe what a vaccine is so we can discuss it intelligently. And that way we can sort of discern fact from fiction. How are they made? What are these controversies and how did they come up anyway? Well, the foreign protein, the antigen, in this case, could be an attenuated or killed virus. So if I were to take a killed virus, say smallpox, if I had a smallpox culture and had some way of inactivating it, either by heat or isolating one that was not pathogenic, and I injected it into you, your body will still recognize the foreign proteins of that virus and mount an immune response accordingly. So that when you eventually get confronted with the real deal, you already have B cells there with antibodies that are going to be all over it and nuke it. You get exposed to pathogens that you previously had been exposed to all the time. It's very rare for a human to get, say, the same cold twice. There are lots of rhinoviruses which cause colds, and presumably with age you should be getting less colds, but there actually are many more cold viruses than you will. If you go through 1 or 2 a year, you know, you'd have to live probably until you were 157 or so in order to overcome it. But the point of the matter is the immune system retains this memory for the antigen. So if we can trick the immune system into thinking it's already seen this pathogen, the next time you actually are confronted with the thing, you're all over it. And that's the basis of what we're trying to do with the vaccine. So and this has entered into the public mystery about vaccines and brings up the issue of, do vaccines cause autism? Well, let me tell you, this is correlation and not causation. So what happens here? What happens here is the mumps, measles, rubella vaccine that kids routinely get about the age of 2. So the parent brings in their kid for their normal MMR vaccine, and the kid comes home and soon thereafter presents autistic symptoms. So the correlation is made, hey, my child was fine before I brought him in, and now the vaccine seemingly causes autism. And this has created quite a media stir, where autism normally can present around that same period of time anyway. It had nothing to do with the vaccine or not. And autism is on the rise, detected in our society, upwards to alarming percentages now. But it's not about the vaccine. And it was thought that one preservative in that vaccine, which contained mercury, could have had something to do with the presentation of autism. This has since in literature been debunked as well. There is less mercury in a mumps, measles, rubella vaccine than you're exposed to in a can of tuna fish. So the correlation epidemiologically has also been established that there is not a causation between the mumps, measles, rubella vaccine and autism. And I'm just going to leave it at that. We could get really a lot into this, but for now. But it is helpful, I think, to understand vaccine production methods. And I'm going to give one example of how we make vaccines in terms of attenuated vaccines. But I first want to show you that there are really basically 6 or 7 basic methods to do this. So if you wanted to make a vaccine, you could, as I just suggested, take a pathogen that is similar to actually the pathogen that causes a disease. In the case of smallpox vaccines, the original vaccine used cowpox, which would cause a similar immune response, so that if someone had been exposed to cowpox while they didn't get the smallpox symptoms, would recognize smallpox when they were infected and mount an active immune response against that before they actually presented any symptoms. So you could use a similar pathogen as a vaccine. You could also kill this live smallpox virus, but then again, some small part might survive and cause symptoms in people that were vaccinated. You can also use what's called an attenuated virus, and that's what's commonly used to make a measles virus vaccine. And I will in a minute go over that, because that is the basis for one of the components of the mumps, measles, rubella vaccine, and I think we should understand how that process is actually conducted. You can kill the virus as what's been done with polio vaccine. Look at it this way. Any of you know anybody with polio? That is almost an extinct ailment. I can see one person. Since the polio vaccine, polio has virtually been eliminated from our population, and I would presume that person is probably older than, say, 50 years old. Actually, for that matter, smallpox was nearly eradicated from the planet. Smallpox was almost extinct, except for small aliquots that were saved, some of which for bioweapons resources, and now we are again seeing a smallpox resurgence. And there was an argument at one point, what should we do with the reservoirs of smallpox? And some of us voted to send it to the sun. You know, what do we need that for, really? But nonetheless, smallpox vaccine has been extremely effective at eliminating smallpox. Another way you can do this is actually take the toxic protein and use that as an injectable to mount a vaccine against, as what's done for tetanus. You can use subunits of a virus, which actually won't cause the disease unto themselves, but allow for the production of antibodies, such as what's done for hepatitis B. And more recently, you can shoot naked DNA into cells, and what will happen there is the naked DNA will come off of a particle, be transcribed into RNA. That RNA will be translated into a foreign protein, and that foreign protein will be acting as an antigen, in which you will now make antibodies. Your B cells will remember that, and then, so there's no virus involved at all. So naked DNA can be designed as a vaccine unto itself. Great idea. Think about that. So in terms of an attenuated virus, what you would do in order to create this, in the case of the measles, is that you would want to have a pathogen that is altered such that it would still invade cells, and you could use those cells to make copies of the virus, just as you would any other live virus. And the altered virus must be similar enough to the original measles virus in order to stimulate the proper immune response, but not so similar as to cause the disease itself. There's a number of ways that you could do that. You could mutate it. You could create a new strain so that you'll need to grow it only in tissue culture under conditions where it wouldn't grow in the human. For example, you could grow it at temperatures that were either too high or too low to actually grow in a human, but you could grow it in the laboratory. This would be an attenuated virus. And you could select for that by routine culture over and over, sort of like an accelerated evolution step on a cultured strain of virus. So then this artificial culture growing in an artificial growth medium for the virus, then you could intentionally make that environment different and use that for a vaccine. So over time, the virus will evolve into strains that will grow better at this lower temperature, and that would be the basis of that attenuated virus. And after that, you would kill that and use that as a completed vaccine. And these live attenuated viruses used as vaccines have been made against mumps, rubella, polio, and yellow fever, and now other types of diseases. And there are other ways to now generate these than simply just growing those in an attenuated media. Now let's now get a little more fancy about this. Those were all viral approaches. If you want to make a bacterial vaccine, it gets a lot more tricky. Look at the complexity of viruses and how they protect themselves, how they're recognized by the immune system is different. And so it's been said that you cannot develop a vaccine against a bacterial pathogen without now knowing its genome. And we've now been able to study bacterial genomes since 1995, and this has aided in the development of bacterial vaccines. Any of you see the movie Contagion? With Matt Damon? If you haven't, I show this in my class. We won't during this class, we don't have time. But I'll just say it's a homework assignment. Go and see this movie. It's extremely accurate. And I'm not a Hollywood-friendly kind of guy. Actually, I diss Hollywood pretty much. I think of it primarily as kind of fast food art. But nonetheless, in this case, this movie depicts a pandemic flu and how it initiates and spreads and what the likely scenario will be and the science that's used to counteract it through vaccine development. This movie is extremely accurate, right down to the equipment they use. The science is accurate, what will happen is accurate, and I would go as far to say that this is not a science fiction movie, not by any stretch of the imagination. And this also does not address, like people might ask, could this really happen? This is not a question, could this really happen? It's more of a question, what will happen when this happens? Because this will happen. We will experience a pandemic. Whether or not it's controllable, whether it goes to the extreme shown in this movie is another question. But we've seen pandemics in the past. And they repeatedly happen by the new evolution of viruses like H1N1, H1N5, etc. And I'll talk about this in a separate lecture. I bring it up now in terms of vaccine development because we experience the flu seasonally and new vaccines are developed every year according to the new flu virus that's presented that the WHO monitors globally. So you notice you go in for your flu vaccine every year. And if you're like me and you're exposed to too many people in a university, it's really a good idea to get a flu vaccine. And you look at this movie and you say, this couldn't happen, right? How worried should I be? Concerned is probably a better way to put it. Pandemics have occurred. And like I said, I'll go into a history of this later when we talk about medical biotechnology. But just for now, the flu vaccine, how is it actually done? Well, there's surveillance year-round into the emerging new pathogens, particularly the viruses that cause influenza, and they're designated by an H number, N number, according to these two proteins that are defining the subtype of influenza virus. It's the hemagglutinin protein that stands for H and the neuraminidase protein that defines the N. And these are highly mutable. And the mutations involved in there result in transmission from avian sources to human or from porcine sources to human, etc. So typically there's a host strain that it incubates in before it jumps to humans, and then a phase in which human transmission becomes more accelerated, and then a further phase in which that becomes pandemic and global. But this occurs. And so the WHO and others monitor this and select a strain which is likely to go pandemic. And that selection occurs during January and March, manufacturing production during January to July, purification and testing July to October, until filling and packaging, and it occurs in your drugstores and CVS sometime in the early fall. And shipping begins in August to November, and vaccination from October and beyond. The question is that fast enough? You look at the movie Contagion and you see what happens if a large amount of this stuff is all of a sudden needed. Look at what happened with SARS, for example, and that outbreak just a few years ago. What could be done to make it faster? Could we make enough? Who would get it first? Hopefully first responders and primary care physicians. This is a question I ask in my class typically at this point in the lecture as everybody's eyes are glazing over. You know, so you've heard enough about vaccines. So how many sexual partners do you expect to have during your time in college? Is that 0 to 5, 5 to 10, 10 to 25, 25 to 50, over 50? And you can see the response by some of my previous classes, owing largely to the lower end of the spectrum, but you can see somebody, well, some people responded in the over 50 range. It's like, Well, anyway. Then I usually ask, well, let's take the girls first, just for kicks. And you can see that everything piles down to the low end of the spectrum as compared to the guys, in which there are more in the upper over 50 range and on up in the boys. But why am I asking this question? Doesn't that seem like too much information for this class? Or maybe it's just flat out none of my business. But actually, human papillomavirus is a sexually transmitted disease. HPV is now known to cause cervical uterine cancer and ovarian cancer in females, obviously. It's also responsible for esophageal cancer in males. So vaccines have been developed in order to prevent HPV infection. By the way, not only is it a sexually transmitted disease, but 26% of college population is infected. That's about right. I usually say 26% of you have it, and I can tell by looking at you, but so do you support a mandatory HPV vaccine? This has been now made mandatory in many states at the age of 12. And some people would say, my 12-year-old shouldn't get this, and there are arguments about this back and forth. And both boys and girls are involved. And a large portion of my student population supports a mandatory HPV vaccine in Rhode Island. Okay, so recombinant DNA technology in biopharmaceuticals, we can use antibodies that target specific proteins. These could include proteins on the outside of cancer cells that are specific to cancer cells. So therefore, you would have a way of seeing the outside of a cancer cell by the proteins that are sticking out of it. That those proteins are unique to cancer cells and are different from proteins in non-cancer cells. If I inject you with an antibody, that antibody could target specifically that epitope, that part of that protein that is unique to cancer cells. This offers up all kinds of possibilities. I could attach a drug to that antibody. I could attach a radioisotope to target that. Rather than blasting you with radiation, I could now target specifically that radiation. I could target that drug. I could stop perhaps cancer cells from dividing more rapidly by targeting certain proteins. So it offers up all kinds of possibilities for cancer, cardiovascular disease, inflammation like rheumatoid arthritis. We can also use antibody-based drugs to modify behavior, proteins and enzymes that are involved in obesity, depression, schizophrenia, and so on. So again, it owes back to this antibody-based drug taking coding sequences in this paradigm I showed earlier. So antibody-based drugs are on the market all of the time. Look at the names of these drugs. Do you think it's strange that they all end in MAB, monoclonal antibody? So all of these drugs that are now efficacious against lots of different diseases are injectable drugs developed on the process that I showed you earlier. So all of them are named after the monoclonal antibodies, targeting, in this case, cancer drugs. So as I mentioned, these antibody drug conjugates are drawing a lot of attention now, mostly because they're specific to their target. You're not just putting a thing systemically through the system. You're targeting a drug specifically at the cells where it needs to be delivered, but also this should lower side effects from systemic small chemicals and lower side effects in general. We can also use magnetic particles attached to antibodies. I actually was involved in some of the early development of magnetic particles. I got a patent at one time that was titled Isolation of Biological Materials Using Magnetic Separation. I don't want to tell you how long ago that was, but you can see how that whole industry now has developed. We made very small nanoparticles that were magnetic. Actually, they were paramagnetic. You can make magnetic particles that are below the range in which they can see each other, so they remain colloidal. And you can attach proteins, RNA, DNA to these, and you can target either DNA, RNA, cells, etc., and separate these out using a magnetic particle. So you could, in fact, take a magnetic particle, put it into a monoclonal antibody, attach it, inject that into a system, and simply put a magnet where you want that target to go. It'll go through your bloodstream and stick right to the magnet, right? What an exquisite drug delivery device. And then later surgically remove the magnet. You can also use radioactivity the same way. You could attach an antibody to a magnet and put it there. You can use quantum dots if you wanted to colorimetrically see something like this. And you can use magnetic particles and antibodies that can be seen in MRIs. So I think we can see in the future, again, I'm interested always in this class in putting on your futuristic goggles, that we can attach small nanocomputers to antibodies. And these will then be circulating in your bloodstream that eventually will carry large amounts of information. I do not think this is science fiction at all, and that you will have information then coursing through your bloodstream that may be possible of detecting, say, GPS signals. Wherever you go, there you are, kind of thing. Oxygen delivery could be occurring the same way, so that there are molecules, for example, that are capable of binding oxygen the way hemoglobin does, but at a km that's much higher. So you can bind molecules of oxygen to these artificial proteins. Think Lance Armstrong on steroids. I didn't mean to say that, but Lance Armstrong on these kinds of steroids. And what you have is the capability of binding slow-release oxygen to the point where humans will be able to walk underwater without assistance. Very cool. Climb Mount Everest without a tank. Jet fighters. And perhaps win a Tour de France or 9. Communications. Is it possible that these small computers will allow me to communicate with you? Very interesting ideas, yes? So antibodies for the future. For now, and I'll get back to that later, about how nanotechnology will interface with molecular biotechnology into the future and some of these kinds of things. And I don't think you should be scared about this, but that's my point of view. I'm more optimistic about technology in general. If someone had told you that you would be carrying 8 gigabytes of information in your pocket 10 years ago, that might have been a scary idea. Okay, rheumatoid arthritis, I think, is the poster child for antibody drug development because of the drug Enbrel and how successful that's been. So what is rheumatoid arthritis? First of all, we have to understand it's an autoimmune disease. What that is you are generating, you remember how I said self is okay, non-self is not okay, until self becomes the problem. And an autoimmune disease is where you are making antibodies against yourself. That is a real problem. So you're making, in the case of rheumatoid arthritis, you are making antibodies against your own TNF factor alpha. That's a protein involved in inflammation. So therefore, the symptom then of rheumatoid arthritis is inflammation. In MS, another autoimmune disease, you're making antibodies against your own myelin sheath and disrupting then that system. So it's an autoimmune disease, and antibodies then can be used to treat it. So here is an autoimmune disease is primarily thought of as an inappropriate immune response of the body against substances and tissues normally present in the body, like TNF, or in the case of multiple sclerosis, your own myelin sheath. Previously, these were treated with immunosuppressants like gold salts, anti-malarials, or a very popular one, methotrexate, or in fact herbal treatments. Methotrexate overall reduces the immune system. So the side effect of that is that, well, it's obvious. You then become susceptible to all kinds of things and often induce remission. So overstimulation of the immune system in rheumatoid arthritis is when white blood cells engulf foreign invaders and make this defense against tumor necrosis factor alpha or other cytokines which are involved in inflammation. So TNF was eventually found to be responsible for as one of the key factors in rheumatoid arthritis, starting with early observations from William Coley through Tony Cerami in the 1970s on tumor formation. And we can recognize that specialized proteins embedded in cell membranes are referred to as receptors, and that these could be involved in recognition of foreign proteins. So in the case of Enbrel, what Enbrel is doing is recognizing the antigen and soaking it up, basically, so that it slows down the progression of rheumatoid arthritis. It also cross-reacts to be very effective against psoriasis. So Enbrel then is a crossover kind of drug that not only treats rheumatoid arthritis, but also treats psoriasis. And other antibody drugs that have been developed treat diseases like hemophilia. Other companies have gotten into the recombinant DNA drug development pipeline. So antibodies and vaccines can now be readily developed through recombinant DNA technologies. And here's some examples of recombinant DNA drugs to treat hepatitis B and acute myocardial infarctions, that is heart attacks. So the immune system then is a very versatile tool to generate new drugs. And rheumatoid arthritis then, as an autoimmune disease, can be treated in this way by generating antibody-based drugs. I want to point out also that viruses, I mentioned the use of particles to deliver antibodies. Viruses are also cell-specific in many cases. Think about HIV seeing T lymphocytes. HIV virus specifically targets T lymphocytes like no other cell type. So in fact, in this way, you could conceive of a strategy in which you could engineer an HIV virus to counteract HIV. If HIV is a good target towards T lymphocytes, can we genetically modify the HIV virus so that it is attenuated, but not only that, that it is suicidal against live HIV? So in fact, using HIV to counteract HIV is a strategy that some companies have embraced. And you can think of this in terms of all kinds of other viruses as packages that could deliver suicide delivery agents to those cells, or otherwise genetically modify cells in what we would call gene therapy. Could we use a virus that would be specific to a pancreatic cell to deliver intact insulin? This has been, on paper, a very easy strategy to conceive, and in the laboratory or clinic, a very difficult thing to actualize. And I'll talk about this in a subsequent lecture as well. Viruses can be used exquisitely for gene delivery. How do you prevent overtreatment, undertreatment, or non-specificity is certainly the problem. So replacement of defective genes, cancer treatments, etc., can all be envisaged using virus particles as delivery agents, but actualizing this turns out to be another can of worms. And viral entry using site-specific receptors to gain entry into cells is certainly a growing field. So we can use DNA-based biotechnology for new pharmaceutical drug development. It's not going to stop with the mere slight examples that I gave you in this lecture. This is going to be a field that will continue to grow. As a matter of fact, like I said, if you're a pharmaceutical industry and you're not on this bus, the bus is already pulled out. There will be subsets of people still involved in small molecule design, for sure. This will not leave. But the intelligence of using specific DNA-based targets can only become more articulated as we get into genomic sciences and so forth. With that, I will close this part of this lecture.